In this video, we are going to talk about DNA repair. You can find written notes on what we're going to discuss today at medicinecanbeeasy.com. The link is also in the description. So what are you going to learn in this video? I am going to explain what DNA repair is and what its basic steps are. We are also going to talk about mismatch and excision repair. I am going to explain how the organism can repair damage to both DNA strands, and we are also going to talk about the difference between non-homologous enjoining and homologous recombination. Okay, so let's start. When we talk about DNA repair, we simply talk about the mechanisms that the organism can use to repair damaged DNA. The organism has different ways of repairing DNA damage, but the basic steps of DNA repair are very similar. First, the damaged DNA must be recognized and removed. This is usually done by enzymes called nucleases, which cleave the phosphodiester bonds between the sugar and the phosphate group so that the damaged nucleotides can be removed. DNA polymerase then fills the gap by synthesizing new complementary nucleotides using the other undamaged strand as a template. Finally, DNA ligase joins the newly synthesized part of the strand with the original one. As you will see, these basic steps are common for almost every DNA repair mechanism we are going to talk about in this video. One of these mechanisms is called mismatch repair. A mismatch is a mispaired nucleotide. For example, here you have thymine paired with guanine that was created in DNA replication. The function of mismatch repair is to repair such errors by recognizing the mismatch using mismatch repair enzymes, removing a short part of the DNA containing the error, and synthesizing new nucleotides with the original DNA strand as a template. Another important mechanism is called excision repair. There are two types of excision repair: base excision repair and nucleotide excision repair. In base excision repair, an incorrect base is removed from the DNA using the basic steps of DNA repair we've talked about. Nucleotide excision repair is used to repair more serious damage to the DNA, such as thymine dimers that distort the double helix. It uses a complex of enzymes that finds the distorted part of the DNA and separates its two strands. The damaged part of the strand is removed and the gap is filled in by DNA polymerase. DNA ligase then again joins the newly synthesized nucleotides with the undamaged DNA strand. So far, we've only mentioned DNA repair mechanisms that can repair damage to one of the DNA strands, using the second as a template. However, if both DNA strands are damaged, the organism can use the double-strand break repair. To be more precise, it can use one of its two subtypes, non-homologous enjoining and homologous recombination. In non-homologous enjoining, the broken DNA ends are just sticked together. As you can imagine, this is not very precise, but it's fast. The homologous recombination is very precise and can be used after DNA replication when there are two double helixes and one gets damaged. First, a nuclease removes the 5' end of the two broken strands at the break. Then, one of the broken 3' ends pairs with the unbroken DNA double helix to use it as a complementary strand. The DNA polymerase elongates the strand, and the strand then rejoins the second broken strand. Finally, the rest of the missing DNA is synthesized and then joined by DNA ligase. The reason we call homologous recombination homologous is because it uses a homologous template. a template that contains the same genetic information as the one that is in the damaged strand. Homologous means the same or similar. Non-homologous and joining does not use such a template, and that's why we call it non-homologous. Okay, so this is it. Thank you so much for watching. If you found this video helpful and you would like to watch more videos on genetics or medicine in general, please subscribe. Thank you again, and see you soon.